Good morning, everyone. Welcome back. Thank you for joining us. We are glad that you're here. I'm Steve.
And I'm Scott.
And together we are the Science and Technology channel on YouTube.
Yeah, and we're talking about a health topic involving Parkinson's. And this is a good one. This is leaning towards the good side.
Very promising.
Yeah. So let's get into it.
An experimental gene silencing therapy safely lowered LRRK2 protein levels in an early Parkinson's trial, but its effect on symptoms remains untested, has produced encouraging results in its first clinical test in people, according to a study published in Nature Medicine on March 24th, 2026. The treatment, called BIIB094 or B094, is designed to target LRRK2, a lot of acronyms here, the most common genetic contributor to Parkinson's disease. Variants in LRRK2 are known to raise the risk of developing the neurodegenerative condition, which affects nearly 10 million people around the world. Would you like to know more?
Researchers have long thought that reducing the activity of the LRRK2 protein might help slow or alter the course of Parkinson's disease. However, turning that concept into a practical therapy has been difficult, and that's according to study co-author Danielle Larson, MD. I'm going to say it's probably 2015, 2018.
I don't know why.
EME. I don't know what all that is. Anyway, assistant professor in the Ken and Ruth Davey Department of Neurology's Division of Movement Disorders.
Quote, this was a multi-center clinical trial looking at an antisense oligonucleotide therapy for LLRK2 specific Parkinson's disease. Wow, that sounded official. Larson said.
That's a word in a spelling bee.
Yeah, oligonucleotide. Continuing her quote, she says, the main goal was to examine the safety of delivering this therapy to patients with the hope that if it proves safe, future studies would evaluate whether it might slow disease progression.
The trial examined whether BIIB094 could lower LRRK2 levels safely in people with Parkinson's disease. Larson said the results indicate that this can be achieved without major safety concerns. The randomized placebo-controlled trial included 82 people with Parkinson's disease, developed across two parts. In the first part, 40 participants received one dose of BIIB094 or a placebo. In the second part, 42% participants received 4 doses of the BIIB094 or a placebo.
So 4 versus 1. One dose of that or 4 doses of that?
Right.
Or the placebo, yeah.
Given every 4 weeks. The therapy was administered intrathecally, meaning it was delivered directly into the cerebrospinal fluid through a lumbar puncture. Cerebrospinal. So that's got to be somewhere probably in the neck area, right?
Yeah.
Brain, spine.
Going right in, yeah. Crazy.
Okay. Kind of dangerous.
In the second part of the study, participants were grouped according to whether they carried a known LRRK2 genetic variant. The treatment was generally well tolerated across both parts of the trial. Adverse events were common, but most were mild to moderate and did not prevent dosing. According to the study, no serious adverse events related to BIIB094 were reported.
In addition to its safety findings, the study showed strong evidence of BIIB094 In addition to its safety findings, the study showed strong evidence that BIIB094 affected the biological target it was designed to reach.
Good.
Cerebral spinal fluid analysis found that LRRK2 protein levels developed by as much as 59% in treated participants.
Yeah, it dropped 59%. That's really encouraging.
Yeah,
that's good. Those reductions occurred whether or not patients carried a known LRRK2 mutation, Larson said, suggesting the therapy might eventually be relevant to a wider Parkinson's population, not only people with a confirmed genetic diagnosis.
This is a quote. The antisense oglio, oglio, olig,
Sound it out.
what did you say?
Oligonucleotide.
Oligonucleotide was designed specifically to reduce LRRK2 expression. That's according to Dr. Larson. Because overactivity of this protein, kinase,
Mhm.
is thought to be part of the problem in Parkinson's disease, including LRRK2 levels could be protective and potentially modify the disease. That's another quote according to Larson. And they said these results point to a possible way LRRK2 targeted therapies might act on the disease process itself rather than other treating symptoms, or only treating symptoms, sorry.
Larson emphasized, however, that the trial was not intended to test whether BIIB094 improves symptoms, cognition, movement, or disease progression. Larger studies that follow patients for longer will be needed to learn whether lowering LRRK2 levels leads to meaningful benefits. In other words, give me your money.
Okay, then this is another quote. The next step would be a phase 2 study with a larger group of patients, according to Larson. Going on with the quote, instead of focusing only on safety, those trials would look at efficacy rather than therapy, whether the therapy can slow disease progression using motor assessments and standard Parkinson's rating scales.
Larson said the findings represent an important move toward targeted treatments that address the underlying biology of Parkinson's disease, and they highlight the promise of BIIB094 and other precise medicine approaches.
Okay, this is another quote. This is one of the first antisense oligonucleotide. Geez, what is it again?
Oligonucleotide.
Oligonucleotide, or ASO. Or why didn't we use that to begin with?
Yeah, I don't know.
Therapies in Parkinson's disease to have safely and tolerability data, tolerability data, according to Larson. It really paves the way for others ASO-based treatments to be developed, potentially targeting different biological pathways. So
this looks promising.
It does, but if I had it, and I'd want somebody to be jumping on this thing because it's a terrible disease. So anyway, but they're working on it, so hopefully they'll get something done on it.
Yeah, sounds promising. All right, folks, thanks for joining us.
We'll see you this afternoon. Thank you.